All right, good evening, afternoon, whenever you're watching this. I want to do something to help you read the Hsieh et al. Paper. And so what we're going to do is we're going to sort of two birds with one stone here. We're going to give you a reading guide, beginning of the reading guide for Hsieh et al. We also have the questions that I've given you for you to work through. I'm going to show you exactly where some of those questions are answered. You want to be able to put together the figures and the text. You want to be able to look at a figure and then find the text to understand what's going on with the figure. And that's what I mean by reading it. So you want to have the paper open somewhere, and you want to pause things and you want to confirm. Oh, can I find this part of the paper? This is how I said things. How does Hsieh et al. Actually say things? And find that part when you can go back and forth and you can read the paper, see what they mean by it. Skip over the parts that you don't necessarily need to understand, but the main gist of it, if it's important enough to be in a figure, it's probably important enough for you to understand. And in fact, supplemental material has a few things that are important as well, but I'm going to show you what those are. I'm going to put them in terms of the things that we've talked about. So I'm going to actually, one of the things I like to do on the test is I like to actually give you a phrase from the paper or a figure from the paper. And I want to just basically say, what does this mean? A, B, C, D. But you tell me what it means precisely, not what it kind of means, especially if there's a better answer for what it exactly means. And sometimes I'll just throw in a not, to change it to the exact opposite of its actual meaning. Look out for that. Be able to read the actual words and to put them together with the figures and the reasoning that they were able to show. These proline mutations make for a more stabilized spike protein that made for a better vaccine. So just to go back to finish up, remember that we showed you this thing about crystal structures and cryo-EM. Because you don't have to crystallize, you can get cryo-EM, but sometimes it's lower resolution. And this is a stark example of what the different resolutions are going to look like for a large bacterial microcompartment. It's not a virus, though it looks like a virus, but it's the same kind of idea. It's a protein shell, symmetric subunits coming together. Very cool. So here's the way, and this is what you're going to be able to look at for the data. For example, this is a channel protein that was found with cryo-EM. It's hard to find channel proteins with crystallization. It can be done, but it's hard. It's easier if you don't have to crystallize them. And so, for example, this was the TRPV1 channel. And basically what you do is you look at the different things. They have these scans that show you sort of the certainty relative to the resolution. And if you have to pick a number for what the resolution of the whole structure is, you pick something like right in the middle of it. And you say that this resolution is probably around 0.25 angstroms. Pretty good. And that tells you where most things are, not as good as a 2 angstrom sort of resolution. By the way, when you have like different lines that are very close together, the difference between these doesn't matter to you. These are different computational methods that they use to solve the structure. Their main point is both of these look the same. The really useful thing is if you look down at panel F. Panel F shows resolution by region of the protein. So overall you have maybe a 0.25, 0.3 angstrom resolution. Where they broke it down by region, and this shifts the resolution numbers around a little bit so that you always have like 3.0 or worse for this. The main thing is to compare this. It's blue where it's 3.0 angstrom resolution, and it's red where that region is 5.0. And what you do is you can see, oh, the top of this protein is all blue. That part I actually trust. The bottom of the protein might be moving around. They might not really know where everything is. There's more error where there's lower resolution with a higher angstrom number. Remember, that's how the whole structural field works. Oh, and by the way, I need to do this a different way because I forgot the resolution. I should have looked at the axes. The resolution is actually 1 over angstroms here. That means that this actually averages out to be about 4 angstroms, which makes perfect sense because 4 angstroms is the average resolution down here. So I take back what I said about what the resolution was. I need to look carefully at this. 1 over what 0.25 is the actual resolution. That ends up being 4. So this is a 4 angstrom resolution structure. That also fits with what I'm seeing. When I look over here, you don't see perfect side chains. You just see general ideas of where the side chain is going to be. So cryo-EM does have strengths and weaknesses. Can measure any size of protein, no crystal needed. As usual, big ones are harder, but they're possible. It can see proteins in the membrane. It can be faster or easier to arrange proteins. For example, the spike proteins were found by cryo-EM, not by crystallography, definitely not by NMR. They're not even close to the size that would work for NMR. The weaknesses are the equipment is not as common, although these days it's pretty much caught up in some places, like there's cryo-EM labs over at the UW. Atomic resolution can be difficult to achieve, but the computations can be biased or constrained out of alignment with reality because it is so computationally intensive. And look at the bottom one. It's very intensive. You're paying a lot for electricity. And these days, electricity is costing more. So I think cryo-EM is probably slowing down now because electricity costs that's how much computing power you need to be able to solve the structure, to put all those two-dimensional things together into one high-resolution three-dimensional structure. But I want you to see this third one. The computations can be biased and constrained out of alignment with reality. If you do some computer checks to make sure that you aren't putting your thumb on the scale and pushing it toward a certain way, but whenever you have that many computations going on, you got to be careful because computation takes on a life of its own, and it starts sort of feeding on itself rather than feeding on the underlying data. And that means that you can be subtly putting in what you want to see. It's a little bit like AI. You put in what you want to see and it moves it away from reality because it gives you what you want to see rather than what's really there. I have to think about what that actually means because it just occurs to me that it's kind of like AI. But the cryo-EM can hallucinate because as you're doing all these computations, you have to do some user input. And you have to say, oh, I think you have an alpha helix here and things like that. And sometimes you can go back, and there are ways to check it, but sometimes people don't run the checks thoroughly. Then other people can go back and they can run the checks. And one scientist did this. So there's a program called Isolde that helps you to refine your structure, but it can also help to give you, you can tilt the structure in the direction that you want. And so when another scientist went in and ran the same program, they found that the map does not appear to fit the model. This is the electron map, and this is the model that they built into it. And you can see all these places where it doesn't fit at all. And there are several examples of people online that have done this and said, something's wrong here. It can actually happen with anything, but with cryo-EM, it's particularly because they move so fast, they don't always they know the checks to do. But it takes time to do the checks. It takes energy and electricity. Maybe you don't even have the budget. So let's realize that actually cryo-EM might be unreliable. The higher resolution, the better you feel about it. But this can happen. It's important. Okay, so now let's talk about reading Hsieh et al. And I just want to show you these things. Pause it, flip to it, and try to find the part of the paper where it talks about it. The first thing that you want to do is you want to look through the news article and read that like a regular article. And you might want to look up some things about what we know about the spike. Basically, the spike is big and floppy. What it has to do is that it changes conformation in a huge way and brings two membranes together for the virus to infect a cell. So it's very flexible, very hard to get the exact structure of. Also covered by a lot of sugars, but we haven't talked about sugars yet, so we're not going to worry too much about that, except you see all the blue? That's actually sugar. So there's a lot of sugar on it as well. Oh yeah, this is a better picture of it. This is the whole spike protein. It's actually a trimer. There's 3 things involved here. If you look carefully, you can see red, orange, and yellow-orange it's a stalk. It's not a very stable, it's definitely not a globular protein. It's a membrane protein. It's just very flexible because it has to be. It's a hook that changes its shape and brings 2 cells together by changing its shape. Here's a picture of the architecture of the entire virus. And there's the spike on the virus surface. The virus actually does have a membrane. That's the case for coronaviruses. And they have these arrangements. You can talk about the arrangements of the RNA inside it, the ribonucleotide. There's tetrahedrons inside it. But what we really care about is all these spikes on the surface. You get an idea of how big they are and how flexible they are. That's the idea. Before fusion, they look like the structure that you usually see. But then after they fuse, they change shape to become from a flail shape to this needle shape. That's a huge conformational shift when you go from the structure on the left to the structure on the right. But it's the same protein. And that shows you how flexible it is. The other thing about before it infects the cell, that's when you want to bind it. That's when you want to attack it. After it affects the cell, that conformation is too late. You want to form antibodies to the pre-fusion conformation. But this is one of the reasons why it's so hard to form antibodies to the pre-fusion, because you don't always see it. Sometimes it's flexing to be a different shape. What they did with the prolines is they locked it into that shape, into the flail shape, the pre-fusion RBD, which is receptor binding domain, pointing down in this case. So this is the pre-fusion spike. That's the post-fusion spike. That shows you exactly the secondary structure changes, tertiary structure changes, the whole thing changes. And the whole problem is, how do we get it to freeze it into the pre-fusion? We can just take the protein and put it into a vaccine. That's great. We get some pre-fusion conformation. But we really want to tilt it to the left so that there's more pre-fusion conformation. So the antibodies are going against the thing that they will be able to block binding before it happens, not be able to bind it after it's only bound already. So here's a gel, and this shows different mutations that they've made. This is kind of a boring gel, because what they're showing is they're showing that there's the size that they want and nothing else. And all of these mutations are just amino acid mutations, one or two of them. In the context of a 1,000 residue protein, it's not going to make any difference on the gel. You shouldn't be able to see very much difference between what's up and what's down here. The other thing you can get from this is the size of the band, not the location of the gel, but the bulk of the band, the color in the band, because that shows you how much protein is there. So you can see that this A942P protein is the same size on the ladder, but it's got a darker blue to it. That means there is more protein. Just making that one mutation ended up getting more protein. They're showing you actually some of the cryo-EM pictures of all the molecules sort of jumbled around. This is where the cryo-EM takes its two-dimensional slices from before putting them together. And they're showing you they tried a bunch of different mutations. Look at what they tried and why and what worked best, what didn't work. Be able to interpret those kind of figures. So ultimately, after doing the cryo-EM, they got a different structure. They got a structure of something called HexaPro. You have to look up in the paper for what is HexaPro. In fact, why not pause it right now, look through for HexaPro, look through the beginning of the paper to find out where they define it. And that will help you understand what it is. This is, by the way, beyond the vaccine version. This is a super stabilized version beyond the vaccine stabilized version. That's a little hint, but they find the trimer. This is the actual the electron density map right here. This is the secondary structure of the same thing. You can sort of imagine how this green part fits into the electron density over there. Then they zoom in on a couple of residues. These are places where they mutated and they put in prolines, so they can kind of see what the proline is doing to sort of hold it at a particular frozen angle. This is a particular picture. This is from supplementals, but it's nice because it shows the different steps of cryo-EM. You don't need to know all the technical details, but you just need to know how this fits with how we described it in class. You have all the different 2D images. You put them together into a reconstructed 3D image. And this is the graph that you want to look at. This is the graph. For this one, they actually don't give you 1/angstrom units. They give you regular angstrom units like I'm used to. And you can see that they're comparing different kinds of different mathematical reconstruction techniques that they used. And if you look over here, you have the different colors, but you can also see that this shows you not just what the resolution is for the molecule, but it shows you if like half the molecule has a worse resolution than another half by this technique. And so you see that this blue and green technique, you actually have, this is a particular conformation where two of the receptor binding domains are pointing up. And the overall resolution is 3.2. This is one conformation with only one of the RBDs pointing up. And this resolution is 3.21. And you can see that they're pretty much similar. They did some mathematical things, but when they did some mathematical processes to get the resolution better, to refine the model, and they ended up like correcting their model until they ended up with like 3.2 angstroms. And just look where it is. 3.2 angstroms on the purple line is right about at the middle, or actually it's right where this blue line is. This blue line is where they say, where this blue line meets the purple line, is what they say the resolution of this structure is. And remember, lower is better. So you can get an idea of the whole molecule is sort of following the same pattern. When you see the blue and green lines over here, you have one part that has sort of trouble with resolution, bad resolution, and then another half of the molecule that is much better. But they actually use that in their computations to be able to smooth it out. Basically, this is percentage of the molecule at that resolution, and this is, you expect this to show this sort of S shape. And then they do one of those maps like I showed you. Instead of red and blue, well, they're still using red and blue, they just throw green in instead of white here. But they're showing you where the different resolutions are located on the molecule. And so you can see here the red areas are 15 angstrom resolution. They really have no idea what's going on in the red areas over here. The blue areas, they do. But you've got to orient yourself. Okay, where are the receptor binding domains? You go back here and you look at this structure. You look at the other structures. I don't show you the figure where you figure out where the receptor binding domains are. But you can, they're part of this right here, and they're the receptor binding domains. They can be up or down. This is what you want to look at the paper to try to figure out, what does this mean? 2 RBDs up. This is a different conformation, 1 RBD up. These are very similar resolutions when you get down to it, but they distribute differently among the different proteins. And so you can look at this. The one on the left is the 2 RBD up, lots of red. The one on the right is the 1 RBD up, much less red. And you can see, where does it have the worst structure, where does it have the best. And then they have this in a table form rather than a number form. And I just want to, there's only a few of these numbers that are really important, but you see they're comparing 2 HexaPro proteins. They got one configuration with 1 RBD up, and they got another one with 2 of the 3 up. Okay? And when you look at them, a lot of the numbers are very, very similar. And even the resolution is very similar. And we can say that the resolution looks about the same. But then we have some things that you can look for. So you look for 4 things that start with R. Resolution, 2 RMSDs, and then Ramachandran outliers. So if you look at RMSD, remember how we had RMSD in the lab when we did an alignment? That's just a measure of how distant are, when you align 2 things, how distant are the same atom from itself. Between the 2 things, you compare each of the same atoms, and you basically get how far away they are. You can do it for a bond and see how far it is from the ideal distance. You can do it for the angles, all the angles in the amino acids, and you can see how many degrees off it is. And so you can actually see that these are very small numbers, 1 degree, 0.008 of an angstrom for the RMSD for bonds, but the smaller the better. And if you're comparing 2 things, the one with the smaller RMSD is the preferred structure. Ramachandran, we talked about that, and they have 95.8% of the Ramachandran angles are in the favored region, 4.1% is allowed, and only 0.1% is an outlier. Probably good. You know, this looks pretty good. But again, you want to be able to compare 2 things. There are some other things that are outliers and scores. Usually you can say that outliers should be small, scores should be, well, you need to know about the score, whether it's a low score or a high score that is good. These are really good for the technicians in the field, but you can tell what the 4 R's mean. And that's how I want you to look at it. So look at the paper with this in mind. This is how to interpret the structures. And some of the questions on the study guide have to do with this. Some of the questions have to do with background. And this is real life stuff. So look up stuff. If you find out something, you can actually, what you find out is real. And so you can look at real sources if you have questions to try to figure it out. That's the way it's going to be when you get a job using this information. So I want you to practice that now. Mostly I want you to be able to pick up a paper and to be able to find your way through it. Not to read every word and understand every part, but to understand the most important parts. What did they do? Why? How did they show it to you? Are there parts that look better of their argument and parts that look worse? That's when you really get into science. All right, so have at it. In a little more than a week from when I'm recording this, we will get together and we will answer those questions on the study guide.